Welcome to this explainer. Right now, our world is facing an absolutely massive agricultural challenge. As global temperatures keep creeping up and rainfall becomes more and more unpredictable, securing our food supply against severe drought isn't just some hypothetical goal anymore. It's a critical, real-world necessity. Today, we're unpacking a fascinating piece of research that takes us from the macro-level threat of climate change straight down into the microscopic machinery of plant cells to reveal how a single gene might just help future-proof our crops. It all really boils down to one fundamental question. How on earth do we engineer our crops to survive on a much, much drier planet? The stakes literally could not be higher. But before we can even think about editing genomes or building supercrops, we have to look closely at nature to see how certain plants naturally survive when the water just vanishes. What biological secrets are they hiding in their DNA that we can learn from and, more importantly, amplify? To figure that out, here's our roadmap for today. We'll start with the global crop threat, then look at discovering the Osr40c1 gene, engineering plant drought tolerance, taking a look inside the cellular machinery, and finally, the chromatin remodeling defense. Okay, let's dive right in. Section 1, the global crop threat. Take a look at what happens when you expose different strains of indica rice to 7 straight days of severe drought. On the far left, you've got the IR36 strain. It's lush, it's green, and it's basically shrugging off the stress. But then, look at the MTU1010 strain over on the right. Under the exact same drought conditions, it is totally withered, rolled up, and brown. It's a striking contrast, right? And it demonstrates something really vital. Nature already holds the biological blueprints for surviving a drought. Some plants just inherently know how to fight back. The real question is, what exactly is that IR36 strain doing differently on a molecular level? Which brings us to Section 2, discovering the Osr40c1 gene. When researchers dug into the roots of these plants during a drought, they looked at transcript abundance, which is basically how loud a specific gene is shouting. And they found a direct, quantifiable correlation with a gene called Osr40c1. In that highly tolerant IR36 strain, the expression of this gene skyrocketed by a massive 7.3-fold. The moderately tolerant IR72 strain saw a respectable 4.5-fold bump. But that dying sensitive MTU11010 strain, barely a 1.9-fold increase. So the data here is crystal clear. The louder this Osr40c1 gene gets, the better the plant survives without water. So what actually is Osr40c1? Well, it codes for a carbohydrate-binding lectin protein. Now, the scientific community actually already knew that this specific family of proteins responded to things like salt and osmotic stress. But, knowing a gene wakes up during an emergency is very different from knowing how it saves the day. The exact molecular mechanism, the actual survival strategy, remained a complete mystery, at least until this pivotal study mapped the whole thing out. All right, Section 3, engineering plant drought tolerance. And this brilliantly illustrates the power of genetic engineering. What happens if we just give vulnerable plants this genetic superpower? When you compare wild-type, just normal, unedited plants, against overexpression lines, or OX lines, the results are honestly staggering. These OX lines have been engineered to continuously pump out high levels of Osr40c1. And the researchers didn't just test this in rice, they actually expressed it in tobacco plants too. Under drought stress, the wild-type plants in both species are wilting, yellowing, and collapsing. But the overexpressed lines? They are lush, upright, and visibly thriving. This proves the gene isn't just along for the ride, it actively drives survival across totally different plant species. And the numbers back up exactly what we're seeing visually. When you summarize the physiological traits under drought stress, the wild-type plants suffer from a really high percentage of rolled leaves and critically low relative water content. But on the flip side, the plants overexpressing Osr40c1 managed to maintain incredibly high water content and totally resisted leaf rolling. They even stockpiled higher amounts of protective compatible solutes like proline and sugars. But that still leaves us with a huge question. How is one single protein pulling off this massive physiological rescue? Let's find out in Section 4, inside the cellular machinery. To understand how it works, researchers tagged the protein with a green fluorescent marker to track exactly where it goes inside the cell. Notice that really strong green glow? It isn't just floating around randomly. It localizes precisely in the cytoplasm and, crucially, straight inside the nucleus of the cell. Now, the nucleus is the command center. It's the vault where all the plant's DNA is stored. So the fact that this protein rushes straight into the nucleus during a crisis is a massive clue about what it's actually doing. It's going right to the source code. But once it's inside that command center, OSR40c1 doesn't work alone. Through a yeast-2 hybrid assay, researchers found out that it acts almost like a project manager, specifically recruiting an elite biological response team. We're talking about proteins with names like OSAM2, OSAP8, OSMEN-B1b, and histone 4. Don't sweat the complex jargon. Just think of them as a highly specialized emergency construction crew. You've got stress responders, DNA-binding proteins, and histones, which are literally the tiny spools that DNA wraps itself around. So the crucial point is what happens next, and a Western blot analysis reveals it beautifully. Under normal, well-watered conditions, OSR40c1 is just a single protein weighing in at 53 kilodaltons. But when drought strikes, bam! The band completely shifts all the way up to 174 kilodaltons. This is physical, undeniable evidence that under the stress of a drought, OSR40c1 grabs those specific partner proteins we just talked about and physically snaps together with them to form a massive emergency multi-protein complex. And get this, if you silence any one of those partner proteins, the plant loses its drought tolerance entirely. They absolutely must work together. And that brings us to the climax of our explainer. Section 5. The chromatin remodeling defense. Why does this massive multi-protein complex form, and what is it doing inside the nucleus? To understand this defense, we have to understand something called chromatin remodeling. Inside a cell, DNA isn't just floating like loose string, it is tightly wound up around those histones, like thread on a spool, into a structure called chromatin. When it's wound up that tightly, the genes hidden inside cannot be read or activated. Chromatin remodeling is the dynamic modification of this physical packaging. It literally unwinds the DNA, allowing dormant, hidden stress defense genes to be exposed, turned on, and transcribed by the plant. So let's walk step by step through this brilliant biological defense mechanism. Step 1, the soil dries up, the plant senses drought stress, and triggers a rapid spike in OSR40c1 expression. Step 2, the OSR40c1 protein rushes into the nucleus and links up with its elite team to form that massive 174 kilodalton complex. Step 3, this emergency complex interacts with the histones and physically remodels the chromatin, unwinding the tightly packed DNA. And finally, Step 4, those previously hidden downstream drought defense genes are activated, flooding the plant with the protective solutes it desperately needs to survive the drought. As the authors of the study summarize perfectly right here, their findings cleanly delineate this totally novel role of OSR40c1. It connects the macro-level survival of the whole plant straight down to the micro-level machinery of epigenetic regulation. By acting as the key that brings these specific regulatory and chromatin-associated proteins together, OSR40c1 is essentially the master switch that allows the plant to unlock its own deep genetic defenses. By showing us how we can harness a plant's own epigenetic master switches, this research just blasts the doors open for the future of agriculture. Think about it. If overexpressing this one lectin protein complex can teach entirely different plants, like rice and tobacco, how to survive a severe drought, we absolutely have to ask, what other dormant biological mechanisms are just sitting there, waiting to be unlocked in the DNA of our crops? The blueprint to securing our future food supply might already be written. We just needed to find the right key. Thanks for joining me on this explainer, and keep questioning the science that shapes our world.